Everybody, we are taking a look at this transparent brain model. Let's see what we can see. We'll go over some structures that you hopefully already know, and then we'll get into some deeper structures of the brain. All right, so let's start with bones. You can see this thicker plastic in the back here. This is the posterior side of the head. You see this little protrusion here? This is the external occipital protuberance. If you feel the back of your head, that little bump that you have, that's what that is. And that's because you've got ligaments that attach all the way down to these spinous processes, like of the C7 vertebra. This is your occipital bone. So this thicker plastic is representing bone. So this is the occipital bone, the external occipital protuberance. This next bone down is the atlas, C1. And so atlas is balancing the occipital bone so that you can nod your head yes. Next bone down, that's C2. We see that it has a spinous process. We see that it's bifid. C2 is called axis. And the special thing that axis has, it has this little dens, I don't know how you can see it, but it's this protrusion upwards that helps you spin your head yes and no, helps atlas spin. Oh, don't mind that. C2, here's C3, C4, C5, C6, all have bifid processes. And then C7. C7 does not have a bifid process. It protrudes out the most. You can feel it on the back of your neck, this bump right here. Once again, it allows for a ligament to attach from the external occipital protuberance down between these bifid processes and attaching to C7. If you've experienced whiplash, you'll feel pain because of this stretch to this ligament. We can see that these cervical vertebrae have transverse foramina for the vertebral arteries that feed the brain. These are the vertebral arteries. We also know that it has a vertebral foramen, vertebral canal that allows the spinal cord to pass through, as well as intervertebral foramen, or plural foramina, for these spinal nerves to pass out of. The hole leading into the skull, that's the foramen magnum. So this is the border between spinal cord and medulla of the brain. All right, so those are all the vertebral and skull features, skull bone features. Let's take a look at meninges. The most superficial protective covering of the brain is the dura mater. The dura mater is just deep to the skull, so this is bone. Here is dura mater, represented in this more opaque plastic. Dura mater has two layers. You've got the periosteal layer, which is closer to the bone, and you've got a meningeal layer, which is deeper. Sometimes they're fused together, but sometimes they separate, and you get this passageway which fits in a blood vessel. This is known as a dural venous sinus. It's a vein. Sinus just means large vein. So this is the dural venous sinus. It drains blood away from the head, drains blood away from the brain. The next layer down of the dura mater, next layer deeper, is the arachnoid mater. The arachnoid mater is a web-like meninges, and it has a subarachnoid space so that CSF can flow. That subarachnoid space will drain, and we can see extending from the arachnoid mater, we can see these little protrusions going into these dural venous sinuses. These little protrusions are known as arachnoid villi. Villi mean little finger-like protrusions. You can also call them granulations, but villi make more sense to me. Arachnoid villi, they're for draining CSF that's full of waste or, low oxygen and full of waste, so we can eliminate that CSF and circulate fresh CSF into the brain through the choroid plexus. Arachnoid villi help drain CSF into the dural venous sinuses. Directly against the brain, the third deepest layer of meninges would be the pia mater. Pia mater is very thin, very delicate. It goes with every single gyrus and sulcus of the brain. So we can't really point it out. We just assume that it's directly against the brain or it's directly against the spinal cord. So once again, pia mater, arachnoid villi from the arachnoid mater, dura mater, dural venous sinus. Now getting to actual brain structures. Those are structures protecting the brain. We can see the lateral sulcus right here. That's this. You can see all the gyri and sulci of the cerebral cortex, this really well-developed part of our brain. If we were to peel open the lateral sulcus, we would see more cortex. That's how highly developed our brain is. So you can see deep to the lateral sulcus, we see more folded cortex. This is known as the insular cortex. The insular cortex is important for a lot of different things. One thing that's important for is for processing smell, olfaction. If we go deep to the insular cortex, we find more brain regions. We're not going to go over this capsule, but I do want you to know the gray matter that we find deep to the insular cortex and also deep to this capsule. It's known as the basal nuclei. When we see it in a frontal cut, we would see it surrounding the lateral ventricle and a bit more lateral of the thalamus. Basal nuclei are important for things like moderating your muscular, skeletal muscle movements so that you can not have tremors. It helps with overall control. If you have Parkinson's disease, you have neural tissue that's degrading in this area. We can see the lateral ventricle right here. If we go on this other side of the brain, we can see the lateral ventricle will continue on to the other ventricles. Let me figure this out. The lateral ventricle will continue to the interventricular foramen, that little passageway here, interventricular foramen, to the next ventricle. This is the third ventricle. The third ventricle has a little hole in it in the middle. It's because brain tissue is passing in between hemispheres. That little protrusion passing in between is known as the interthalamic adhesion. From the third ventricle, we continue on to the cerebral aqueduct. The cerebral aqueduct is at the level of the midbrain. From the cerebral aqueduct, we continue to the fourth ventricle. And then from the fourth ventricle, we continue to the central canal in the medulla and in the spinal cord. So once again, the order of cerebrospinal fluid flow, it gets produced by this pink stuff. That's the choroid plexus. It's found in all the ventricles. Choroid plexus produces cerebrospinal fluid, lateral ventricle, interventricular foramen, third ventricle, cerebral aqueduct, fourth ventricle, central canal. It'll then make its way to that subarachnoid space, and then it'll get drained by the arachnoid villi. That's how we get our cerebrospinal fluid circulating. Other things that we can see here. On the underside of the brain, we see this crossing. That's the crossing point is the optic chiasm. We have an eye here, an eye here. These are optic nerves coming from each eye, and that information can cross over. Optic nerve, chiasm, optic tract. We can see that here. Put our pieces together. Optic nerve, chiasm, optic tracts. And that's located on the inferior aspect of the brain, and when it is a really useful landmark because right underneath, just inferior to the optic chiasm, we can find the pituitary. And just superior to the optic chiasm, we can find the hypothalamus. So I'll show you that again. Right where the optic chiasm is, we find the hypothalamus, this brain region, which let me show you on this side. Optic chiasm, hypothalamus. You can find the hypothalamus right here, just above the optic chiasm. The hypothalamus will connect through a stalk called the infundibulum, and that infundibulum is found right here, connects to the pituitary right there. So here's our pituitary. It's a gland. We'll get to when we get to the endocrine system. It's sitting in the sella turcica of the sphenoid. It's connected to the hypothalamus through that infundibulum. There are neurons that extend back from the hypothalamus to the pituitary. So here's our hypothalamus. You can see our thalamus. You can see our thalamus here. There's that interthalamic adhesion that passes through that hole in the third ventricle. You can see the cerebral aqueduct, central canal. Cerebral aqueduct is in the region of the midbrain. So here you can see the midbrain, where you see the round bulge, that's the pons. The narrower part is the medulla. When you look at the midbrain, one important feature that you can see are these two bumps on the dorsal side. When I say dorsal, I mean this back side of, like, the cerebellum would be right here. You can see these two bumps there. That's the dorsal side. Collectively, these four bumps of the midbrain are known as the corpora quadrigemina, literally the four-bodied twins, corpora quadrigemina. The top ones are known as the superior colliculi. Colliculus means bumps. These are the inferior colliculi. Superior colliculi are important for a visual startle reflex, like if you see lightning and you flinch. The inferior colliculi are important for an auditory startle reflex, so if you hear thunder and you flinch, that's mediated by those regions. Once again, on the ventral side, you can see these bulges. That's the pons. When we're looking at the inferior aspect, you can see the optic-chiasm, infundibulum, leading to the pituitary. These two round things, those are mammillary bodies. They're important for, they're part of the limbic system. They're important for memory. There are lots of nerves, cranial nerves that come off of here, and we'll get to that in another video. When we get to the medulla, we see two rounded protrusions, more lateral. See what's shown there in red? They're rounded, they're olive-shaped, so they're known as olives. These olives are areas of nuclei. Just like we have basal nuclei and other clusters of cell bodies in the brain, these are areas of nuclei. And then these midline projections, these are tracts, or they're axons, white matter. They're known as pyramids. So we have pyramids that go along the middle of the medulla, and we have olives on the lateral portions of the medulla, white matter and gray matter. One other thing that I can show, did I show the basal nuclei? If I didn't, I did. Okay. I think that's it. Let me know if you have questions. Thanks for listening.